Thanks so much. And yeah, I'm happy for you to interrupt me. I'm going to tell you a couple of stories, and it's complex because we're talking about instrumentation as well as the molecular ecology. So don't be shy about interrupting if I'm not clear on things. And yeah, and I kind of restricted it to 2 stories because of the complexity of it, but there are a lot of like really intriguing details within this. So hopefully we could stimulate some conversation afterwards. But I really appreciate the opportunity to talk to you guys today. And I've been doing some work with eDNA recently and a lot on technology, but I am a microbial ecologist, and I really want to, every opportunity I get to talk about microbes, I'm happy for it. So thanks, Rachel, for the invite. So yeah, so we know on the left here, this is a picture I lifted from Jed Furman's website, that microbes are dense. They're very, there's a lot of them in seawater, and they're very diverse. So you can see a diatom here in the picture, and all those little dots are bacteria, and the smaller ones are viruses. And you've got 1 million of them per milliliter seawater. And if you take the volume of seawater in the planet, you can imagine how many and how complex that diversity is. Yep. So there are a lot of microbes in the world's ocean. There are a lot of different taxa. But the reason that Rachel and I are studying them is because they mediate biogeochemical cycling. We know that we're emitting a lot of carbon dioxide into the atmosphere, and the oceans take up about 25% of that carbon dioxide from fossil fuels. And the mechanisms of the biology and how that affects the biology, how the acidification affects the biology, but also their capacity to take up carbon dioxide is going to change as the oceans change. And we know this is happening already. So it's really important to understand their drivers, and that's kind of what my research aims to do, kind of scale up what we're doing for microbial ecology observations in order to get better data to project how they'll change with future ocean change. So here's a really nice synthesis from this year from Browning and Moore, and this is feeds off of some work from Mark Moore in 2013. And what this is the result of a lot of really difficult experiments at sea, looking at the nutrients that fuel carbon fixation. So what you're looking at with each of those dots, it's where there was an experiment, and those experiments typically are looking at chlorophyll, changing chlorophyll as a result of adding nutrients. And the nutrients tested are down here. Well, I've highlighted some of them here, but they're all down here. So as you add iron, phosphorus, zinc, these are combinatorial experiments. So you end up looking at chlorophyll change in response to all of these different nutrients. Here's an example of what these little dots mean. So your primary nutrient that is limiting carbon fixation is going to be on the inside of this dot. And if it's co-limiting, it's going to be divided in half. So here you can see that iron and nitrogen are co-limiting primary nutrients. And then you have, and you can see this here. So if you add nitrogen or iron alone, you actually don't see a stimulation of chlorophyll. But if you add them together, you do. And then the secondary limiting factor is going to be something like cobalt or B12 in this case. And that's what you're going to see on the outside here. So this is B12 and cobalt. So if you add them in addition to those primary stimulating nutrients, you see another increase in chlorophyll. So all of those data and all of those very difficult experiments were performed at all these little dots. And the big take home point here is there are a lot of areas in the ocean that are nitrogen limited. So if you add nitrogen, you're going to see a stimulation of chlorophyll and photosynthesis. And not just that, in this paper, they showed that a lot of areas are also co-limiting. So you're going to see a secondary stimulation of photosynthesis if you add a second nutrient. In the open ocean, there's a lot of, not a lot of nitrogen. And so your sources of nitrogen are primarily deep water, so from the deep water moving upward, or nitrogen fixation. And I'm going to talk about 2 case studies today. One of them's up here in the North Atlantic on the coast of the US, and that one's going to be about nitrogen fixation. And I'm going to talk about some work that I did when I was at UC Santa Cruz with John Zara's group and Chris Schwollen's group up north of Hawaii. So north of Hawaii, we see that you know, in the ocean here, in the vast open ocean, it's actually pretty stratified. It's really warm in the summer. You see this really hot top layer that sits on top of the cold, deeper layer. And so despite that stratification, year on year, there's a bloom. There's a big bloom in the North Pacific. You could see this is chlorophyll on the bottom here. And that bloom is, it almost happens every year, but it's pretty predictable. And so why would there be a bloom when the ocean's most stratified, when there's not so much mixing coming from below? So here's a hint into the mechanisms. These are some data from 2010, but they're a compilation of Argo float profilers. And they go out and measure the physics of the ocean, but this is from Ken Johnson's group, and they added nitrate sensors as well. And you end up with these really dense data sets where they're profiling up and down. And you can see, for instance, here, this is on the edge of an open ocean eddy. So this is the sea surface height. But here's where this data are from. And you end up seeing, like, over time, you see these kind of episodic events where nutrients come into the surface ocean. So this spot right here, this one time was when you saw this event here. And so that nitrate does make it into the surface ocean, and it does make it into the photic zone. And because you've got your nutrients and you've got your light, you could get the photosynthesis to happen. But again, here's, this is basically the density. This is telling you how stratified things are over the course of seasons. So this is the summer months. This is the densest, the most stratified part of the year. Yet you see that these episodic events. So we decided to use these eddies as kind of a target to see if we could see a change in that nutrient profile and a change in the photosynthesis and the microbes responsible for fixing carbon just north of Hawaii. So this device here is an environmental sample processor. I'm going to call it the ESP. And we deployed the ESP on a drifter north of Station Aloha, kind of on the edge of this eddy. It went northeast and then it went northwest. So the environmental sample processor, it filters seawater. It's kind of the Swiss Army knife for molecular detection. It can do everything. It can do in situ processing. That's why it's called a processor. But you could preserve that filter for later analysis. So you could go back in the lab and do whatever you might want with it. But it also extracts cells on board. You could do a ribosomal RNA hybridization to get a fingerprint of the microbial community. You can do quantitative PCR on board. You could do toxin detection. So it's a workhorse for harmful algae in the West US. And it transmits the data in real time. So we used the ESP with Mbari and a load of engineers and scientists that work on the ESP. And we kind of adapted it with molecular ecology tools to look at different things like nitrogen fixation and to look at changes in the community over time, the microbes in the North Atlantic or North Pacific. Now, I'm not going to tell you about most of these data because the talk's too short, but I'm going to tell you about the latest of them. So we took those filter samples and we took them back to the lab, and we did some sequencing with them to figure out what the community is doing. So I showed you some data about nutrient limitation, and these are some other data from Claire Mahaffey from the North Pacific again, and nutrient limitation. And what she did were these deck incubations where you have your seawater and you add in silicate, phosphate, nitrate, and you look over time. And so you basically are looking to stimulate photosynthesis. So you're looking for an increase in chlorophyll over time. So she did these experiments, and these are, I've put in lines for every 24 hours, and it took about 3 days to see a change in chlorophyll, and it took about 3 days to see the nutrients start to decrease. And from this paper, she said that there's a potential risk in extrapolating the response of phytoplankton to these short-term studies, because if you're just measuring over 24 hours, you're not going to see that response. But there are other phytoplankton responses that we could measure, and one of them is looking at their fluorescence. So you could look at the activation of their fluorescence in real time. But what we chose to do is look at their RNA. So here we deployed the ESP again here, and it was archiving samples. So filtering, preserving, filtering, preserving, filtering, preserving at about 25 meters depth, and as it moved along this gradient. So here's basically the transect. And what you see is you see this oxygen profile where, again, it's you've got your deep chlorophyll maximum where you see some oxygen, but the surface is devoid of oxygen, really. And then we happened to see this little nitrite pulse here, and we were on the ship, so we were taking frequent CTD casts. And this one event, this little pulse of nitrite, can account for about 33 to 98% of the primary productivity that we measured in that same region. Whereas the nitrogen fixation rates we measured in that region only counted for less than 6% of the primary productivity. So we did that on deck on the ship. So those are some measurements that we did while the ESP was deployed. Some other things we did while the ESP was deployed were some incubation experiments again, and here we're like listening to the microbes, we're listening to their RNA to see what's really driving their distributions. So this is Kendra Turk and I, and we spent a lot of time doing these deck incubations and filtering the seawater, etc. But the experiment I'm going to show you is using 25 meter seawater and 130 meter seawater. So we took water from those depths, and we're calling the 130 meter seawater deep. It's not very deep, but we know there's more nutrients in it. And what we did was I'm going to show you 3 different treatments. One of them is the control, where we're just diluting that surface seawater with filtered surface seawater. Another one's the deep water mixing one, where we took unfiltered deep water and mixed that with the surface water. And then the last one is filtered deep water with the surface seawater. So what we're looking for is the response of the surface microbial community, for the most part, to the deep water nutrients, but the deep water nutrients with the biology as well. These are the results, and I'm going to kind of walk you through this. It's heat maps are no fun. So the punchline here is each of these columns is a replicate. It's one of the treatments. So we did 2 replicates for the filtered 130 meter seawater. We did 2 replicates for the unfiltered 130 meter water addition. And we did 2 replicates for the surface filtered surface seawater addition. And each of these lines is a gene that was turned on or off. If it was turned on, it's red, and if it was turned off, it's blue. So we have loads of data from the transcriptomics, and they all show us the expected patterns. Now, there's a lot of details here that were unexpected, but I'm going to show you the punchline of the big take-home message. So the transcription profiles are very repeatable. So our treatments looked identical to one another. So we did multiple treatments in different incubators, and they looked the same. But also the mixing treatments, so where we took the deep water and we mixed it with the surface microbial community, those look more similar to one another than they do to the control. So that means that that nutrient response is going to be the same between those. And what we saw specifically was that there was a relief of nitrogen, phosphorus, and iron stress in the dominant plankton that we looked at. Those are Synechococcus and Prochlorococcus. And that with that relief of nutrient stress, we saw a stimulation of photosynthesis, carbon fixation, nitrogen assimilation, the things that you would expect if you saw a response from the phytoplankton. Now, what we did with those deck incubations is we compared those transcriptomic profiles to what we saw in the ESP. So again, the ESP was sitting here at about 25 meters depth. And we took some of the samples from days kind of surrounding this event that we saw in the surface ocean and looked at the metatranscriptomic profiles. And what we're going to do is compare what we saw in the incubations that's here. These are filtered deep water with the surface microbial community, deep water that's unfiltered with the surface microbial community, and then the surface control. So here I've got just basically those pictures. And in the middle here are the ESP samples. And what we found is that the transcriptomic profile of the ESP samples looked very similar to that deep water mixing experiment. So it does look like those nutrients are getting into the surface ocean, that the 25-meter community is responding to those nutrients in similar ways to how they did in our deck incubations. So this tells us that these events are happening, that they're happening despite stratification in the summer months. And there's a lot of doors that it opens up, because if you think about it, is it just happening in the summer? I mean, at least in months where it's less stratified, you would expect it to happen more frequently. And how frequent are they happening within those eddies? So we just looked at some of those samples surrounding that event that we measured, but in reality, we could have looked across the entire eddy, and we didn't do that. So is this a response in all of the surface plankton kind of near the periphery of eddies? How long does it last? There are a lot of questions about the microbial response. So going back to this question, can deep nutrients enter the photic zone during the stratified summer months? We say yes, they can, and they definitely stimulate the phytoplankton in the surface ocean. So yeah, does this happen at the peripheries of all eddies? Does it happen in the Atlantic as well as the Pacific? These are questions that kind of we're exploring now, and there have been some recent studies actually looking into the same. We are going to be out in March near the Canary Islands, and the Canary Islands are northwest of Africa. And there's what they call the eddy corridor. There's an area there where the coastal water basically spins off of the coast of Africa and heads west. And we'll be looking at these eddies with a combination of autonomous vehicles. So we're going to be doing this in March, where we have a sea glider with nutrient measurements and plankton response. So looking at the photophysiology of the plankton. We're going to have Autonaut looking at the photophysiology of the surface ocean, and then we'll have this this device. It's called the AutoSub, and that's at the National Oceanography Center. We're going to have this beast with everything in it. It's a huge vehicle, so we're going to have a genomic sampler in it. We'll have a cytochip. So this is a miniaturized deployable chip that looks at fluorescence and impedance to get an idea of the taxa that are there. We'll look at the fluorometry underway, and then we're going to have some nitrate, phosphate, and silicate measurements, as well as some carbonate system measurements. I work at the NOC. I lead this ocean technology and engineering group, and this here is their workhorse. So this was a program that began with Matt Mollum when he used to lead the group, and they have these little miniaturized lab-on-chip wet chemical sensors, and they kind of mimic what you would do on the bench in the lab. And so they have really high accuracy and really low limits of detection. So they kind of allow you to get into that surface ocean and measure a lot more than you would be able to do with an absorbance, for example, measurement. And then I'll tell you in a minute about this genomic sampler that we're bringing out. Right, so these are some data from Luo et al. This is another synthesis looking at nitrogen fixation. And historically, when kind of we started, well, actually, I'd say just when I started working with John Zare's lab at UC Santa Cruz, the data on nitrogen fixation were primarily in the open ocean. And you could see in the synthesis, you've got a lot of open ocean measurements. Here's a lot of data from Station Aloha. But because of that, there's a bias in nitrogen fixation research that used to exist. So here we're talking about nitrogen fixation as this other source of nitrogen for phytoplankton. And it's typically people would measure it where other nutrients are low, because the idea is if nitrogen fixation is bringing in a new nitrogen source, they're going to stimulate carbon fixation. But because of that offshore bias, you end up measuring very distinct communities and ecologically restricted communities, really. So nitrogen fixation used to be considered to be most important for primary production in open ocean regions. There was a perception that the nitrogen fixers aren't super competitive for other resources like phosphorus, iron, etc., and that nitrogen fixation occurs where dissolved in inorganic nitrogen is low. And Rachel here knows a lot about this because she's published papers on these nitrogen fixers, so you probably have heard about the ecology, but I'm going to just talk about two of them today. Trichodesmium is a colonial nitrogen fixer, and it's filamentous, so you could actually see it in seawater. So that's, yeah, it fixes nitrogen and carbon, and it does oxygenic photosynthesis, whereas Eucine is a symbiont. It fixes nitrogen, but yeah, it doesn't fix carbon or produce oxygen. And there's all sorts of interesting ecological data out there now that John Zare has basically thrown every tool that exists at this symbiosis of Eucine. So punchline is that it's symbiotic with Brarudosphere bigelowii was the first, but with these haptophyte species. And that because the nitrogen fixer is symbiotic, it's lost a lot of genes. So it's lost photosystem 2, so it doesn't do oxygenic photosynthesis. Rubisco, so carbon fixation, it's lost that capability. It's just got this very restricted genome. So you can imagine that changes the ecology of where and when it can bloom relative to something like Trichodesmium that's more capable of doing physiologically diverse functions. And I wanted to say there's some papers down here. These are really good papers. If you have time and you want to read about them, there's a nice review, and then two of the fundamental papers kind of figuring out this symbiosis. Yeah, so this is a study that we did here in the Northwest Atlantic and off the coast of the US. We're looking at nitrogen fixation, and we wanted to kind of figure out about this coastal nitrogen fixation, because it does seem like it contradicts these paradigms. And we went to this specific region because there was a really good paper by Marjorie Mulholland out of in 2012 that kind of showed us that there were really high rates of nitrogen fixation unexpectedly in this region, and that they're on the shelf. They're not way offshore like you would usually look for them. So I'm going to tell you about Roxy. Roxy is a robotic cartridge sampling instrument, and where the ESP is this workhorse, and it's a Swiss Army knife, does all these in situ analyses. All Roxy does is filter, and then it preserves that filter, and you take that filter back and process it in the lab. So because it's much more simple, you could make it a lot smaller, and you can deploy it. We usually put it on the bench if we're at sea, but you could put it in a little pellet case and put it on a buoy or a mooring, or you could ship it out pretty easily. It's a it's an easy device to use, but it was developed in ocean technology and engineering with some EU and NERC funding, and it's now commercialized with McLean in the US. So we had to demonstrate that, sorry, I should say here that we were looking at nitrogen fixation here. So my objective was to get high resolution genomics and figure out what nitrogen fixers are there. And I was working with Duke and with Nicolas Cassar's group to look at high resolution nitrogen fixation rates. So he's got a really cool device that I'll tell you about in a minute. So kind of coupling the ecology to the rates. So here we've got the auto sampler. So this is the data from the Roxy, the device that was compared to our manual collection samples. So each of these letters A, B, C, D that you see in this transit, we went from Bermuda off the coast of the US and stayed in the coastal region on the shelf and then returned to Bermuda. But each of these letters is here, and we compared first the qPCR data using those manually collected samples to the Roxy collected samples, and what we found is a pretty good relationship. It's a one-to-one relationship between those two, so that tells us it's good for quantitative PCR. And then we compared metabarcoding data, so looking at all the 16S and all the 18S ribosomal RNA. And according to this PCA plot, you actually see this really nice relationship. So you see a clustering of EE would be manually and Roxy collected samples at Station E on the coast here. So that seemed to work. And in addition, we were using the Faracus. This is Nicolas Cassar's group's work, and this is cavity ring down laser absorption spectroscopy, and what it does is a acetylene reduction assay, and that gives you a gross nitrogen fixation rate. And they from that cruise, they compared those data. This is a Faracus, that's the name of the instrument, and it's continuous. It works under on a continuous flow system. Compared those data to some incubations they did with N15 substrate, and there's a pretty good relationship. Here's a one-to-one relationship, but in general, Faracus overestimates nitrogen fixation rate a bit. And here's a zoom in on this bit right here. So it does overestimate nitrogen fixation, but at the same time, it's gross instead of net nitrogen fixation, and you kind of expect that with those measurements. And it gives you much higher resolution data than you can do with incubations. So paradigm one, nitrogen fixation is highest and most important to primary production in open ocean regions. So yeah, we went to the same region. We moved along the coast here, and we did find that the nitrogen fixation actually in the coastal region was highest, and it peaked here off the coast. So it seems that was contrary to that paradigm at the time. And we took some high resolution samples from that genomics instrument along the same series. But what Wei Tang found when they were measuring these rates is that the top these represent the top 2% of global nitrogen fixation rates, and they're in a coastal region, which is counterintuitive based on the paradigms, and that in that region at that time, they can contribute up to 40% of net primary production. They're not competitive for other resources. So here are some data on nitrate and nitrite, phosphate and iron, and how those distributions looked. And you could see kind of in the coastal region, there's more phosphate, there's more iron definitely over here, where we saw the peak rates of nitrogen fixation. And there's more nitrate and nitrite in that region. So they seem to be doing very well and being very active where nutrients are highest. And so it seems that they are competitive to other plankton. So in addition to the continuous nitrogen fixation, the NIFH gene quantification, so NIFH is the gene that's representative of the nitrogenase that fixes nitrogen. And that was showing the highest quantification on the coastal area for USNA. So this is similar to what the Mulholland et al. Found. So nitrogen fixation was highest in the coastal region, UCNA was peaking in the coastal region, and trichodesmium was more abundant offshore as was known previously. And similarly, if you look at the RNA, so if you look at NIFH transcripts, you see that not only are UCNA abundant in the coastal region, but they're also very active. And trichodesmium is most active offshore. So that coastal nitrogen fixation is attributed to UCNA for this study. What's driving their distributions? So I didn't get to show you this, but in the ESP study in the North Atlantic, we found that not just in that transect, but also in the summer months and at Station Aloha historically, we saw a really nice relationship between UCNA and phosphate concentration. So it looks like they're kind of driven by phosphate in the North Pacific. And similarly, in the North Atlantic, we saw during this cruise a really nice relationship between UCNA abundances and phosphate concentration. And we found, looking at their RNA in more detail, that the phosphorus stress markers were most expressed here, where you could still find them present, but they're in this region, they're more phosphorus stressed because there's less phosphorus as you move away from the coast. So the last paradigm, nitrogen fixation occurs where dissolved inorganic nitrogen was low. I kind of showed you the punchline here already, but yeah, we had quite a lot of nitrate in that coastal region, which is again where we saw plenty of UCNA and plenty of nitrogen fixation activity. So this is kind of once you know the genome, and due to this hard work from John Zer and his group, it makes a lot of sense. So they're missing the nitrate reductase genes and transporters for using these other nitrogen sources, and that they're missing ammonium transporters from their genomes. So yeah, it shouldn't matter at all what the nitrate and ammonia concentrations are. So is this exception to the rule? How representative was this field study? Well, this was a while ago. So we did this in 2017, and that group had already done a couple cruises in 2015 and 2016. We find that, yeah, it's again and again, you see this coastal nitrogen fixation that's peaked. And then there were some follow-on papers showing that in a lot of coastal regions and really in the Arctic everywhere, you end up seeing UCNA. So these are UCNA counts globally. And there have been loads of papers since. So UCNA is against all the paradigms, and it's not acting as you would expect. And because of this, you know, we need to start thinking about changing our models. So when people adapt Earth system models for things like adding nitrogen fixation, they're thinking about the old paradigms and the old drivers, and we need to really start thinking about the ecology of the nitrogen fixers, but also use things like machine learning, where you might be able to get at those data without actually doing a model for every single ecology. So here are those old Luo et al. Observations for nitrogen fixation, but there was just this year, this big publication where another synthesis, and now everybody's looking in the coast, and it's fantastic. We'll have a lot more data to really show where nitrogen fixation is important. So this idea of really doing this high resolution combination of the physics coupled to the chemistry to the biology, it gives you a lot of insights. When you do this high resolution study, you could see those drivers because you end up seeing a lot more variation. Even though it's a short time frame, you get a lot more data in that short time frame. So you could really use those relationships to kind of plug in and model things better. But you could also just learn a lot about those the biogeochemical drivers and what's going to change with ecological or environmental change in the future. So yeah, with that, I just need to thank all these people. So I'm the one giving the talk, but because you work on technology, it's a lot of engineers involved. There's a lot of ecologists involved, and a lot of people helped at seed. But this is work from MBARI and UC Santa Cruz that was in collaboration with University of Hawaii, as well as the work that I've done here with University of Western Brittany, Duke, and here at the NOC. So I'm happy to take questions.